So Mauve is a way to do a large-scale genome comparison to look for rearrangements in genomes and things like that. It was developed by Andrew Darling, and this is the only place that exists outside of the Mauve website that I know of. The README is there, but let's go into Tools and Services, and let's go down to Genome Alignment. And we click on that. Notice that a B follows it, meaning that it's only for use with bacteria now. But okay, here's some questions. Mala, could they submit a viral genome right now in Mauve? I would think like maybe the pox viruses that are big enough, those might be interesting, but who knows? He says, au contraire, he has used it to visualize viruses and it has been the greatest experience in his entire life. I'm paraphrasing, but I know that's secretly what he meant. So we can add the genomes one at a time. You guys should be up to date on this and it should be no problem at all. Or you can add a genome group. And just for kicks, and just because this is pretty fast, let's look at our Ricea group. I know it's kind of boring, but okay. And add that. Now, one of the advantages is if you add these genomes individually, is you get to select the reference genome that's on top and the ones below it. So if you're doing something for a publication, and you notice I say that a lot, because the only way you survive in science is publishing. And so I'm trying to get your mindset around creating a story here. You're hopefully not a fiction writer, but a fact writer, but you're creating a compelling narrative to the reviewers of your work. And part of that is the illustrations. So I want you to be always thinking about that before you submit that article, that your illustrations are the best. And just for this test, when I submit a genome group, it's going to randomly pick what the reference genome is and then the target genomes. When you add these individually, the first one that you add is the reference genome. And then the others are the comparison genomes, and it will keep them in that order. So once you look at it the first time, you may want to make decisions as to who you have as reference or comparison genomes. So we need the folder, which is that one. We need the output name, Ricea ANL alignment. And then if you understand all of this stuff that is part of Mauve, knock yourself out, play with that, do whatever you want. This is one place where I don't mess around with it and I submit the job. Ding. If we click on the jobs icon here, we can see it's running. Generally, this is pretty fast, but you never know how long it's going to take. So let's go into workspaces and go into BV-BRC workshop. Click on genome alignment. Click on, for this one, we'll do Ricea. And then click on that checkered flag. And so you can see a lot of files here. I don't care about any of those. But up here is my eye candy and it's screaming to you, look at me, look at me. So we click on that and let's hope it loads. If not, it's going to be another belly flop off the high dive. Still loading. So this is the reference genome. And what this is doing is it's breaking up the reference genome. It's looking across and looking for large components. When Bob showed this tool to you, he was looking at a gene neighborhood. This is an entire genome. And so it's making a gigantic gene neighborhood and looking for areas of similarity across this group. This is the BRHL group, which is from the human louse. Here's my beloved gorilla louse. And what's the difference that you see between these two genomes right away? One is big, one is much smaller, which is an indication that this guy has been throwing things off right and left compared to this guy. So the gorilla louse is not as needy as the human louse. So this endosymbiont, there's a lot of things that's just thrown off. What do these big red lines mean? They're the indication of contigs. Here is a chimpanzee one, and then another chimpanzee one. And look at this. So it's showing this big section, and when you mouse over it, it's showing you where all those things are. And so it's showing that this is at the end of this genome and at the beginning of this genome. Now, it could very well be that these are just de novo assemblies and the contigs are in no particular order and they are not reflective of what's happening in the actual genome, or it could be that this is actually a rearrangement that has occurred in this genome. And so you can see all the way down here, and you can see that some of these have more contigs than others. You can also start zooming in and in and in and in. And sometimes when you zoom in, you can see that other colors pop up. This is something that's in the gorilla genome that was not in the human one that was used. So if we keep on going in and in, then you can also get a sense of the genes that are in that particular area. So look, this is a tRNA synthetase gene, and you can just mouse over those and see what they are. And if you were interested in one of them, you could click on it, but this would give you a link to that gene, and it would open the feature page. And then you can go back to see the whole thing. So it's a pretty simple tool, but actually this is extremely popular among biologists. You used to see it a lot more than you currently do, but Mauve has been a standard tool in the biology toolkit for bacteriologists for a long time. And I have another tool there. Click on Genome Alignment and then Brucella. I've talked a lot about Brucella in here. And so Brucella actually have 2 chromosomes. These are all the reference genomes, and it's very well known that the abortus strain has this rearrangement here that the other strains have in a different orientation. So it was discovered actually using this particular tool when they looked at that long, long ago. And the Brucella genome is actually in 2 chromosomes that are reflected here. What they think is that the second chromosome was actually a plasmid that they picked up at one time and then have since embellished upon it and brought in more data. And now it's in 2 chromosomes. But this is the kind of way you can use this tool. So it's very simple, very neat. I really like the visualization of it. The one thing that I really dislike about our version of this tool is it does not give you a download SVG function, which I think is a big failure. And I think we put in a ticket for it. So I've actually had people who want to use it in a publication. And with great shame, I told them, well, you'll have to take a screenshot and submit it that way. But both you and I know that that's not going to pass the quality that they require for publications. So I told them the way I had to do it was I would take an image of it, and then I use Illustrator. I would upload it in Illustrator and I would trace the object and then try to make it as neat and clean as possible. But that is the weak sister to an elegant, elegant design. So hopefully the people in the back row are listening to what I say, and they are assigning a ticket right now to enable that. Any questions before we head into the fireworks display at the end? It's RNA-seq. This is exciting. I want to see it. Skunk pox, vole pox, raccoon pox, tetrader. Is that tetra pox? What is that? A turtle thing? What is tetra? Somebody look that up. Vaccinia, which is the smallpox one. Cow pox, camel pox. Oh, Malik, you have succeeded beyond my wildest dreams. That is fascinating. Look at that. Isn't that cool? I want to go in and see what these, well, looks like there's a, oh, look at that. Oh, that's just fabulous. How much fun. I'm a little worried about these genes that are floating off in space. Oh, I can see that there's something here that in the visualization is a bit wonky when I dig into it, but still, it does work. Yes, question. You cannot search for it, but you can find it. You can get a sense of where it is, because this will tell you the position on this particular contig. And so if I know that, I can go into the feature page or the protein page for any of these genes, and I can see where that gene is. And I know it's on this contig around this position because it gives you a start and stop. And so using this, I'm able to find it. And then there it is, the anchorin protein, just where I wanted it to be.